Okay, great. We are happy to have you here. Thanks everybody for joining us today. So I'm going to introduce Dr. Arseny. Dr. Arseny Finkelstein is an assistant professor in the Department of Physiology and Pharmacology, School of Medicine, Tel Aviv University. He interestingly started working on developing immune-based therapies for stroke and other neurodegenerative disorders, but later he switched to studying brain functions under normal physiological conditions. During his postdoctoral training at Howard Hughes Medical Institute, he studied brain circuits involved in working memory and decision-making and developed novel optical methods for manipulation of neural activity in the brain that you're actually going to see it in their recent Nature papers. So now the floor is yours.
Yeah, well, thank you very much, first of all, for the invitation, and I'm happy to present this work and would be interested in hearing your thoughts. So, any questions during the talk or after, yeah, I'll be happy to have it as interactive as it gets. So, the research, my general research interest is how does the brain generate behavior? How does it generate cognitive functions? We focus on decision making and the use of short-term and long-term memory in guiding decision making behaviors. And what I will focus on today is how cortical interactions on the level of the cortical microcircuitry within the motor cortex are organized during naturalistic goal-directed behavior. So if we think about the relation between the function of the neurons and their organization in the brain on different scales, we of course know about local interactions on the level of the microcircuitry, connectivity between individual neurons, but also interactions on a large scale system level between different brain regions. And while we do know quite a bit now from anatomy about the pathways in the brain on this large scale level, we know quite little about how local connectivity on the level of the microcircuitry is organized. What are the connectivity principles? What are the statistics of connections? Who is connected to whom in terms of the functional identity of the neurons? And on the other hand, we have these beautiful representations that we can refer as tuning curves in the visual cortex. We have orientation tuning. If you go to high order areas like hippocampus, we have more complex tuning curves such as place cells. And somehow these interactions on local and a global scale give rise to these functions. And if we want to study, for example, the mechanism of the formation of orientation tuning, we can drive these neurons rather easily by presenting sensory inputs and then study the local circuitry and the large-scale circuitry organization. But in order to map responses in this high order brain regions, in other non-sensory regions, we need essentially animals' cooperation in driving the neurons. So in the case of place cells, you can let the animal run in the environment, and you would have this beautiful place cells. But if you go to motor domain, to behaviors that require some movement and also decision-making paradigms, you often need the animal to learn some task in order to exert this movement so that we can map the responses of the neurons during this movement generation. And this typically, as I don't have to tell this audience, this requires quite a bit of shaping and training. And basically, this animal learns to do this this reaching task, and then we see the corresponding representation, the directional coding in the motor cortex, or a movement, eye movement related coding. Let's say if we record in a visual, in a fixation eye movement task. So basically, we need the animal to learn some task in order to map their responses. And this learning might change the underlying connectivity during the learning process. So we essentially learn some potentially learned connectivity structure. So in order to try to see, and this is sort of one of the goals in our lab, to see how tuning emerges from connectivity and how it develops during learning, we wanted to have some kind of baseline task that would not require animal any extensive learning, and then yet would allow us to map its motor repertoire. So that was our starting point to this paper. So what we did, and that was inspired by the primate literature on saccadic eye movements and reaching tasks, we wanted to have essentially multi-directional goal-directed behavior in the mouse, and we use motorized water spout. So the mouse is head fixed. It is sitting in front of this motorized spout, and it licks it for water reward. And the spout can arrive in different locations on this grid of possible positions. We use different sizes of grid, in this case 5 by 5. Basically the task is very simple. So imagine I have this water bottle and I just grab it with my tongue and collect the reward. And then on the next trial, the water bottle appears in another position and I again reach it. So very simple task, but it gives this multi-directional movement repertoire. And mice become engaged in this task and essentially perform it from the very first moment of head fixation. It takes them about 10-20 trials to understand that there is water, and then they would just perform it, and they would do hundreds of trials essentially without any pre-training. So this, we believe, opens a window into studying essentially in rodents to map the behavior. This, we call it naturalistic behavior just because it doesn't require extensive training. Map this behavior in relation to neural activity and connectivity, as I will describe. So we do, in this paper, we used 2-photon calcium imaging in order to map the neural responses. We also do neuropixels in our lab. In this case, we focus on motor cortex, specifically on the anterior lateral motor cortex, which is in mice is the orofacial motor cortex. There is debate whether what kind of motor cortex is it, premotor cortex or not. These divisions are sort of fuzzy in mice, but we know that inhibiting this area prevents leaking. And in more sophisticated tasks, inhibition of this area prevents, erases short-term memory for target locations that the animal licks based on cued behavior. So this is a sort of a high, we believe it as a high order area. By the way, the mouse can lick also without this area, but its memory for target locations would be severely impaired. So in this behavior that we did, we can map the activity of neurons. So we do calcium imaging, and if we focus on a particular neuron, let's say this neuron, during this task and look at its activity trace, we would see oftentimes differences in responses to different target locations. So we can trial average the activity over many trials, and then we get this maps. This is a mapping of the response of one single neuron in the motor cortex during this task. We see this neuron is active during the presentation of the target in the central upper part of this grid. And in this case, as you can see, it is also active relatively late. So it is active after the initiation of the movement. And I should say that this is a calcium trace. So this is a bit a slow response also due to the kinetics of the calcium. But even if you take this into account, we see many neurons that are active, not necessarily during the movement, but also neurons that are active after the movement. And this is one neuron. So if we just ignore its temporal profile and just focus on where in this space it is active, we can make this compact representations of its response. So this neuron, again, is active in this upper part of the central part of the grid. This is one neuron, and this is just example of 100 neurons that we recorded simultaneously in this task. In fact, we record many thousands of neurons simultaneously. This is just an example. And you see many different neurons have different responses. So this is our neuron. There are also neurons that are tuned similarly to it. So there is this neuron that also prefers the same location. There is this neuron that also prefers the same location. There is this neuron, and there are other neurons. So this neuron, for example, prefers the upper left corner, and again, it has partners with similar tuning. Some neurons have a sharp tuning, like this two. Some neurons have a broader tuning. So together, it seems that in this behavior, we see essentially a map of this target space in the motor cortex of this location neurons. And one of the hypotheses that we tested whether neurons with similar tuning would be preferentially connected to each other. So this representation of essentially a map of the target space, we think of it as some analogy to the cognitive map for in the context of navigation. So you can draw some analogy between these cells and the place cells in the hippocampus. And I'll show you why we think of the cells as sort of a cognitive map later on. Any questions?
None so far. It seems like we can continue. Thanks for asking.
All right. So I showed you the responses of the cells to target location. But in fact, in this task, we were playing also with the reward size. So on 80% of the trials, the animal was getting what we call the regular reward. And in about 10% of the trials, we were either increasing the reward or omitting the reward altogether. And turns out that these cells are sensitive to the reward that the mouse gets. So now I'm not showing you the location tuning of these neurons. I'm just focusing on their temporal response, on their PSTH as a function of reward size. And what you see here, for example, this neuron is increasing its response following large increased reward and is decreasing its response during reward omission. This neuron number 2 behaves kind of similarly. We also have neurons with different tuning. So this neuron is increasing its response to reward omission and decreasing its response to large reward. There are neurons that are suppressed following reward omission, etc. So we have all different combinations of responses to reward, and about about 20 to 30% of the neurons care about this reward modulations. There are also neurons that are sort of inactive during regular, and in this case also reward omission conditions, but are sort of emerging only upon a large reward. And now if we do take into account their location tuning, and this is the expansion of cell number 1 and cell number 2, we see that basically reward modulation sort of acts, in this case, as a gain of their existing location field. So these neurons can be thought of conjunctive neurons encoding reward location and the outcome. And as you notice, these neurons are also active after the movement. So one hypothesis that we have, and this is something that we are studying now in the lab, is that these neurons might encode essentially a vectorial reward prediction error, which might be a more efficient mechanism for learning. So in this case, these neurons essentially tell you about what was the action and what was the outcome of this particular action, hence vectorial, not only just a general signal of whether you did good or worse than better or worse than expected. They also encode the identity of the action, in this case, the target location. Now, in this experiment, we're imaging the motor cortex. We are imaging it also volumetrically, so different slices in the axial, in the z dimension, which allows us to study essentially the three-dimensional anatomical organization of these cells. And if we map these different cells based on their preferred direction, so basically we sort of neglect the fact that this neuron is a 2D neuron and just look at its directional tuning within this 2D field. So this new neuron would be tuned upwards. We can then look at the different distribution of the neurons based on their directional or location preference in an anatomical space. And what you see is that it's kind of disorganized. But then if you focus, you see that the neurons appear to be on a local level somewhat clustered. So here you see this cluster of green cells. Here you see this cluster of purple cells. Here you see this cluster of orange cells, etc. And when we quantify it, we see a pretty clear distance-dependent organization of this tuning. So this is the tuning correlation, how much the tuning is similar between neurons, and this is the distance between the neurons. We see that nearby neurons are tend to have a similar tuning. And if we decompose this distant dependency into the lateral dimension and the axial dimension, the three-dimensional, the depths between the neurons, we see that the lateral similarity in tuning drops faster than the axial ones. So that means that neurons tend to have a more similar location to neurons that sit above or below them than neurons to the left or to the right of them, which suggests that basically this similarity in tuning is following an organization into mini columns. So this is the motor cortex, again, of the mouse in this task that doesn't require training, which we think it reflects sort of the the baseline state of the motor cortex. So basically, most of the work that I described was done during my postdoc in Karol Swoboda's lab at Janelia, and the focus of the work in our lab in Tel Aviv is basically studying the changes in the neural coding and the local connectivity as we transition from this basic behavior, from this baseline state behavior, into more complex behaviors, in this case, behaviors that require a probabilistic inference and decision-making among multiple alternatives, and track the changes in the network, both in terms of the neural coding and the underlying connectivity. So what I'll show you today is basically this part, the relation between behavior, connectivity, and coding, and I'll happy to talk about this part now or on a different opportunity. So in order to map connectivity in the living brain, we used an all-optical approach using targeted optogenetics. And of course, understanding the relationship between tuning and connectivity is a major big question, and people attempted to do that and mapped this relationship, typically using slice work or paired recordings, which gives real connectivity between neurons, synaptic connectivity, but it's very low throughput. And this result suggested, for example, like-to-like connectivity in the visual cortex between similarly tuned orientation-tuned neurons, the work from Tom Mercic-Fluge's lab. So here we took an approach based on calcium imaging and optogenetics. And what we do here, we stimulate a target neuron optogenetically, and then measure the responses in connected neurons. We define connected neurons as neurons that basically turn on upon stimulation of the target neurons. And we measured this responses in the network, again, using calcium imaging. So we do calcium imaging, we stimulate a neuron, and if some other neurons light up in response to this neuron stimulation, we call them connected neuron. In this method, and we do this very fast, we cycle through many different targets in a random pattern, and that allows us to map many potential connection pairs in a very short period of time. So we do essentially mapping of about 500,000 pairs of neurons in this volume in about half an hour. And this allows us to create this sort of maps of, we call it causal connectivity between neurons, causal functional interactions on the level of the microcircuitry. What this connectivity maps, again, is essentially suprathreshold responses to the stimulation of the target neuron. So we are only able to map sufficiently strong connections that would be above the spiking threshold of the postsynaptic neuron. So this is a big sort of a big limitation of the method. But on the other hand, we believe that because of trial-to-trial variability, we sometimes have neurons, even with weak connections, that this stimulation brings it sufficiently close to cross the threshold and respond. Another limitation that this method does not map necessarily monosynaptic connections. We know that some neuron was turned on after the stimulation of the target neuron. We don't know whether this response is direct or indirect. So when we map the responses, the relationship between the connectivity strengths and the tuning similarity of this location coding neurons that I showed you before, we see that essentially a like-to-like connectivity between similarly tuned neurons. So neurons with similar location tuning tend to be connected more strongly to each other than neurons with different tuning. So essentially, we see a like-to-like connectivity. Now, just to show you how it looks like. So this is a target neuron. We activate it. This is trialed average of many activations of this specific target neuron that occur at different random times during the experiment. And this is the response of 3 other neurons that, based on the statistics, we define as causally connected to this target neuron. So you see that upon stimulation of this target neuron, also these 3 neurons change their response. These 2 neurons respond in activating, in increasing their activity, and this neuron actually shows inhibitory response to the stimulation of this neuron. In this experiment, we only image and photostimulate only excitatory neurons. So essentially, any inhibitory responses between 2 excitatory neurons are mediated by polysynaptic chain. We do a lot of control experiments showing that these responses are not caused by just indirect photostimulation of the direct stimulation of this follower neurons. We do it using control targets and measuring the responses to them. So I already showed you that neurons with similar tuning are arranged in this kind of mini columns just based on tuning similarity. And I showed you that neurons with similar tuning are also preferentially connected to each other, this like-to-like connectivity. Now, in terms of the anatomical organization of this connectivity responses, we see a strong distant dependence of the connectivity, of the connection strengths, and actually also connection probability on distant dependence. So neurons that are nearby are strongly connected to each other. This response, this connectivity strength decreases over about 100 microns, and then is reversed by inhibitory response further away laterally. So essentially, we have this functional column of excitation followed by lateral inhibition, and the connectivity is stronger within this column axially in the z dimension compared to laterally. So again, neurons that are above or within this column are more preferentially connected to each other, and they are also share similar tuning. So basically, this like-to-like connectivity in location tuning exists within a functional mini column of excitation and inhibition. And this connectivity motif essentially resembles a Mexican head of excitation and inhibition over anatomical space. So this were general statistics of the anatomical connectivity profile. There's many columns of excitation inhibition and like-to-like connectivity motif between location tuning neurons. But in fact, when we look at these maps, we see very complex response connectivity patterns between the different neurons. So each neuron is represented here by a dot, and here we see different arrows at difference. The color of the arrows is indicating the connection strength between neurons, and the size of the neuron indicates how many postsynaptic cells this neuron is connected to. And what we see if we measure the probability of a cell to have a certain number of connections. In this case, we measure outgoing connection so basically how many cell this particular neuron is connected to postsynaptically. We see that most neurons in our mapping are connected to a few cells, about 10 cells, but some neurons have a surprisingly large number of connections. So these neurons here have 50 neurons to which they are connected. And this distribution of connection probability is very far from what you would expect from a random network with the same average size of connections as the one that we observed. If we would just maintain an average number of connections, we would have that most neurons would only have a small deviation from this average in a random network, but we see some very non-random connectivity distributions. And these neurons that are rare and have exceedingly large number of connections, we refer to them as hub neurons. Now, what could be the role of such hub neurons? So if we think about, let's say, the airports in the USA, if you want to go from a particular city to another city, and you don't have hub airports, you would need to move from hop from one a city to another and have essentially a pretty long pathway in order to reach your destination. On the other hand, if you have hub airports which are connected to multiple destinations from a particular city, you can just go to the hub neuron and then go to your final destination. So basically, hubs in a network shorter the path. In this case, the information flow in the network. So we looked at the relationship between hub neurons and their positional tuning, the tuning of neurons to different target locations. So we asked if a particular neuron has few connections, like this neurons here, or many connections, like this neuron here, what would be, on average, its positional tuning? Would it be sharply tuned to the location of the water spout, or would it be broadly tuned? And we found that neurons with actually fewer connections have sharper tuning to location compared to neurons that have many connections. So essentially, hub neurons are not strongly tuned to location. So they have weak positional tuning to reward location. And I'm not showing the plot but it's in the paper, and I'm happy to go also to the actual PDF if you would like. We also see that hub neurons encode the movement timing somewhat more strongly. And they also have higher activity correlations to neighboring neurons. So basically, hub neurons are have more connections to its neighbors and are more correlated with them, which may imply that essentially hub neurons are sort of, if you want, maybe some kind of pacemakers of local network activity. Their connectivity profile allows them to play such a role. So we think that while these hub neurons are not exactly strongly tuned to location, they might, and this is a speculation, they might be the ones that are responsible for the timing of the movement. So you essentially select the timing of the movement using the activity of the hub neurons, and this movements at a specific timing can be directed to different target locations, which the less connected neurons essentially play a role in selecting. So this is a speculation that we have. In the paper, we also included a network model. And if there is interest, I can go over it as well by my colleague, Ran Darshan. And in this model, we suggest that essentially hub neurons might, although they are weakly tuned, but still they have some tuning. And actually, the tuning of the hub neurons is correlated to the tuning of their postsynaptic cells. And because they are connected to a lot of neurons, they can essentially act as amplifiers of the tuning in the network. So instead of having many less connected cells that would have to have stronger like-to-like connectivity to essentially get amplification of the tuning, you can have less of this like-to-like connectivity of the less tuned cells, but more of hub neurons that would introduce, due to their spread, essentially a tuned component to the network, which would act as an amplifier, similarly to what you might have in attractor models of feature selectivity. So, so these are sort of different speculations about the possible functions of hub neurons. So to sum up, what we have a new method for mapping, connectivity mapping, and I should say that we are sort of standing on the shoulders of giants here, people that introduced targeted optogenetics, a laboratory of David Tank and Chris Harvey and Carl Dysert and many others. And we essentially scaled up this approach to have to map a large number of potential connection pairs. We have this new task that allows to map the responses in the motor cortex without training, and this task allowed us to find target location neurons, essentially a map of such target location neurons, a network of cells encoding target location, but also the outcome of specific actions. We show a like-to-like connectivity between this reward location neurons that is arranged within functional mean columns of excitation and inhibition. And if we zoom in into the specific architecture of the network, and this is something that we are only starting doing, we see some neurons that are highly connected to other neurons, have a different tuning statistics. They're more weakly tuned, as I mentioned, these hub neurons. So as I said, one speculation that they might be a pacemaker of the motor cortex. So yeah, so that's basically covers the highlights from this paper.
Thank you.
Yeah, I'm happy to have any questions, discussions, and hear your thoughts.
Yeah, thank you very much. Very, very interesting. Thanks for the talk. I can start asking the question. So, going back to your initial results showing that the tuning of the neurons like kind of smoothly changed, but more laterally. I was wondering, just remembering the range of the distances were around like 200 micron or something like that. So I was wondering whether that's all within one area with one kind of cytoarchitecture, and also if you kind of move the microscope a little bit away, so you have a boundary of an area here, would you expect to see the same smooth transition, or you would expect to see like a large change suddenly when you go through that border between the areas?
Yeah, so that's a great question about the scale of this connectivity or similarity in tuning. The range that we see, this 200 microns or so, it's actually something that a number that repeats itself also in other studies that mapped the relationship between connectivity and also function. So you see in visual cortex, for example, in terms of the connectivity statistics, if you measure this is work both from Tom Mercich-Flugel's lab and some other related work measuring connections. You see essentially a sharp drop in connection probability 100 or 200 microns away. And if you do simulations of the cortex, you just put the morphology of the cells and the density of the cells we are, by the way, I didn't mention that, but we are mapping layer 2-3. So we are looking at pyramidal neurons, and just their geometry suggests that you would have, even if the connectivity would be completely random, it's much more likely to find partners next to you from the overlap of the axonal and dendritic trees. And because of the sort of columnar organization of the cortex in terms of the direction of the axons, and neurons that are on top of each other would have a higher connection probability. So we think that this sort of statistics is something that you would expect but from the geometry of the cortex. But of course, we know that certain areas have distinctive boundaries. If you go to the barrel cortex, you have transitions between barrels, and some other areas have distinct boundaries. We are now we have mesoscale imaging of larger fields of view spanning from these frontal areas all the way posterior to high order visual areas. We don't have connectivity mapping, but we can map activity at cellular resolution. And there we see even we have more distance, so we see even larger distant dependence spread, and this distant dependent similarity. In this case, we measure similarity in just correlations of the responses, regardless of tuning. They are area specific. So there are some areas, all areas show it, but some areas have it a more extended, and some areas, actually, barrel cortex is one of them, have it more narrow. So yeah, so I would expect some distinct boundaries, at least in some areas, but not necessarily at all.
Thank you very much. We can go to
Andrew.
Yeah.
Oh, great. Thanks. Really, very interesting talk. I was very curious about these hub neurons because a lot of us are focused on these other class of responses that is often seen in the primate motor cortex, which is the so-called condition-independent activity. This you may be aware of, but basically, if you look at the population level activity in the motor cortex during reaching, there's a very strong, let's say, target-independent, condition-independent response that happens at the transition, basically from posture to movement. That there's not any obvious reason why that should have a particular anatomical substrate, right? I mean, this is something that is based on many neurons projecting onto a particular dimensions of the neural state space. And I'm just kind of wondering, do you think of a connection, an explicit connection between these hub neurons and this kind of condition-independent response? And more specifically, because that's the other thing I'll let you respond, is just to say, did you look in the paper in terms of the temporal dynamics of the hub neurons during the licking? Because in the primate reaching, they generally have very specific temporal. Well, that's how they're identified, with very specific temporal dynamics where they increase at the round of movement onset, and then they kind of go away during the reach itself.
Yeah, so we are looking at temporal dynamics quite a bit with calcium imaging. I should say we have a course way, but we also do without connectivity mapping. We do neuropixel recording, so we kind of know what to expect from these neurons in the motor cortex, not necessarily hubs. We see basically in 90% of the neurons in the motor cortex, they are tuned to movement timing. So if you forget if you call it direction selectivity for simplicity, a lot of neurons would be modulated at some point during the trial. We expected, based on work, including what I worked on in kind of trained animals, in cued memory-guided cue tests, that the activity that we will see a lot of preparatory activity. Most neurons would fire before. We actually don't see that. Most of the cells fire after, and this is not related to the calcium. This is sort of beyond the calcium time scales. And sort of going back to the literature, we saw that indeed in tasks where the animal was sort of less trained, there was a higher propensity of these sort of late responses. And what we do now is doing longitudinal experiments when we start with this sort of naive task and just measure the responses over time. Because despite the fact that I said that the task doesn't require training, there is some learning that happens in terms of reaction time and other parameters, and then train on a more acute task. So to characterize the system, we see a lot of this responses, not necessarily at the exact transition from to movement, but probably reward related. But some neurons do have preparatory dynamics and this shift also on the population level in terms of the geometry. You see sharp transitions. Hub neurons, what we saw, and one of the problem is that they are very rare. So you sort of getting statistics on them is hard. That they seem to be sort of more had sharper temporal tuning than the average neuron. So, but I can't say whether these neurons at what phase of the trial they are more active. As I said, the majority of the neurons are active kind of after the movement.
So they have sharper tuning. I mean, I know this is a little bit of speculation, I guess, but just so I understand it, your impression is that they have sharper tuning, but that the temporal distribution of that sharper tuning isn't necessarily, not obviously, let's say, early in the movement. If
So for the temporal distribution, we don't have enough of the hub neurons to have a reliable sort of
Yeah.
ordering of the preferred times. And yeah, that's one thought. And another sort of direction that we are going into is into tasks that even this task has essentially a queue, which is the arrival of the link port at different locations. So the movement, although there is no explicit go queue, and the animal can take its time if it wants, there is a go queue. So we are interested in also characterizing the system in spontaneous movements that are sort of entirely self-generated without cues. Maybe these neurons would have a different role there.
Yeah, thank you. Very interesting.
Yeah, I can ask Ali a question. Thank you very much for the talk. Very interesting. I had two questions. One of them may be smaller. Maybe I missed it, but did you look at the hub neurons, whether they provide excitatory or inhibitory responses to the neurons that they're connected to? Or is it just a mix of them, both inhibitory and excitatory, based on the principle that you showed for the connectivity?
Yeah, so I kind of skipped this this part, but the inhibition that we can detect is extremely weak. So I showed you this kind of Mexican hat of red and blue, but actually, if you look at the scale of the blue, is kind of 1% of what we see in terms of the excitatory responses. So we can detect excitatory responses much more reliably than inhibitory responses. And one of the major reasons is that, as I mentioned, we are activating excitatory neurons and recording from excitatory neurons. So every inhibition has to go through an intermediate inhibitory neuron, essentially polysynaptic chain. For the analysis of the hubs, we only focused on the number of excitatory connections. We don't have in this setting a way to answer that.
Thank you. Yeah, I haven't. I mean, I think you kind of answered my question. The other one was that the Mexican hat that you showed, you have like excitatory nearby and then inhibitory on the sides. Does it also sort of like continue? Because when you have inhibitory on the excitatory neurons that are further from you, so you technically have excitatory for the neurons that are further from you. So it's like more like a sync function, but if it's very small inhibition, maybe not that much because it dissipates in the distance.
So in this Mexican hat maps that I showed, basically this analysis is done by centering the response on every neuron and then averaging its profile over anatomical space. So in that sense, beyond the initial sort of column of excitation and inhibition, of excitation and lateral inhibition, you basically almost don't have any connection probability. But there would be other neurons with sort of similar profile further away of excitation inhibition. So this is sort of neurocentric view of the organization. I hope it answered your question.
Yeah, thank you.
Armin. I have two questions. One of them is kind of more detailed. So you mentioned that the hub neurons have a higher correlation to the neighboring neurons. So in my mind, I was thinking that that could be actually the opposite for hub neurons. You would expect that if they have a general role, if they have a condition-independent role in controlling the dynamics and controlling the behavior, you would expect that they also have a high correlation to the neurons that are far away. And you kind of also mentioned about that's the role of hub neurons. So that's kind of contradictory into my mind. And how do you exactly see that?
Yeah, so that's a good question, and I'll happy to sort of clarify it. When I say, so all the analysis, including the analysis of the hub neurons, were done essentially by looking only at responses within 100 microns. Because of what I described as a drop in connection probability, we wanted to limit in order to make the comparison between different neurons. Essentially, we look at a given volume and ask within this volume, within this radius, three-dimensional radius from the cell, what is the connection, how many connections it has. So we kind of equalize this number between all cells. So when I say that hub neurons had higher correlation than non-hub neurons to its neighborhood, I mean within this 100 microns. There could be that there are some other postsynaptic connections that we miss, or they're just too weak, and maybe we miss those. But for the sake of this argument, the increase in correlation to the neighborhood suggests that basically a hub neuron, the more connection it has, the more correlated its activity is. We can't establish a causality here, whether these correlations are because this neuron is driving all of its postsynaptic cells, and that's what increases the correlation in the neighborhood. Of course, there are some recurrency, so it's we are not trying to make this separation. But at least it goes with along this argument, and I should say that this finding that their hub neurons are more strongly correlated to the network is done on an independent measurement from whether a hub neuron. So we are not doing it during the connectivity mapping when we stimulate the cells. We do it during sort of another epoch of spontaneous activity when we don't do any optogenetic stimulations. So these two findings are sort of independent.
Yeah, thanks. Just a quick follow-up on that. Maybe I missed it. So are these neurons more correlated with the neighboring neurons that are in the same, in different depths, but in the same laterality, or is it more, are they more correlated to the neurons that are like in different lateralities, essentially?
So we didn't parse it. We only looked at essentially a volume defined by lateral depth, lateral distance, and the four imaging planes, which was about 120 microns by 120 microns sort of cube around the cell.
Right. Yeah. Thank you.
I can ask another question if you have time. Do you guys have questions? No. Yeah. Okay. Oh yeah, I wanted to ask about this sort of, if I summarize correctly, you have these groups of neurons, a column that has a tuning to certain directions, and then they sort of excite each other and then inhibit the nearby neurons that are tuning to other directions, if that's correct. And then if that's correct, what does it mean for the population activity? Do you see that when the mice reaches to a certain location, that those neurons that tune that location start exciting each other, and the other neurons that encode other locations starts getting inhibited, or is that not what the network as a whole does? Just wanted to see your thoughts.
Yeah, so we're that's something that interests us a lot. So in terms of the model, a simple model of action selection that could actually be consistent with potential continuous bump attractor models that were proposed for place cells, for example, and we tend to think of this system, this is a hypothesis we are testing in similar ways, that you might have essentially a bump of activity corresponding to the target location that would form through this like-to-like connectivity between similarly tuned cells. And in this type of networks, you would have essentially inhibition of neurons that have a different location than where the bump is formed. And the degree of this inhibition can be variable based on the specifics of the architecture. That's on the level of a model. And such mechanism, essentially such cooperation between similar tuned neurons, inhibition of other neurons, might implement a sort of winner-take-all dynamics, which you may want to have when you need to choose a particular target. You don't want your sort of movement to be indecisive during the actual motor production. Because we are limited by the extent by how much we can measure inhibition, we only saw like-to-like connectivity with this gradual of the connectivity strengths as a function of the like-to-like similarity. So basically, similarly tuned neurons have stronger connectivity between them, and the less connected you are, the more you close to zero. This curve actually does cross the zero, so very far away neurons with opposite tuning seem to be slightly inhibiting each other, but it's sort of very close to the zero level. And because the inhibition is measuring it is problematic, as I mentioned, I sort of don't want to claim that indeed there is strong inhibition. We can measure it through different ways, but not in this experiment. Yeah.
Question?
Yeah.
Hi, Arseny. Thank you for that talk. It was really cool.
Hi, thank you.
So the task that you were studying is essentially a two-dimensional planar task for the animals. They have to lick one of several grid locations in a 2D grid. But of course, there's a third dimension, which is depth, forward and back. You had them all at the same distance. So I guess I wanted to get your thoughts on how that third dimension might be coded in this scheme, and whether you think, I was thinking about it, more like a radial coordinate system might actually be what's encoded here, where direction is sort of, angular direction away from the mouth, plus a distance, elevation, azimuth, and distance. So what do you think about how a third dimension might be incorporated into this scheme?
Yeah, so I have to say that in my PhD, my work was on studying a three-dimensional representation of head direction cells, and also three-dimensional coding of other spatial variables. So I'm sort of very intrigued by this question, and I'm ashamed to say that we didn't map it, and I can only speculate based on just observing the mouse behavior also during sort of natural licking. We have in this tuning, although we see clearly neurons that are tuned to different vertical elevation of the target, this tuning is less pronounced than to the left or to the right. You can see it through when you do decoding analysis and also quantification. There are clearly cells that have beautiful tuning to different corners, but it just not sort of seems to be that important feature, at least in this brain area. And when we observe the behavior, actually the mouse, it doesn't try that much to target its tongue to reach different locations of the tube because of the extent of the movement, they tend to just exert the tongue sort of almost all the way down. And then on the way back, they sort of collect the water reward. So they can essentially solve this task without aiming that much vertically. And I suspect that this is also a general feature for for this orofacial licking behavior. You kind of you I don't know, when I see my dog licking, it just exerts the tongue all the way down. But in terms of the dimension that you were asking, I think actually there, protruding the tongue along the rostrocaudal axis, I think this is a much more sort of specific and directed movement. So, retrospectively, I think maybe a more interesting plane would be, left, right, and sort of further away from the center. But we didn't map it. We actually have the motors motorized in all these 3 dimensions. Maybe for a future study.
Okay, thank you.
Thanks, Arseny. I might usurp my power to ask you a final question, a quick one, like an opinion one. And that's about the beautiful small wordness result that you showed. So that kind of a small word connection, it makes a lot of sense to me when you think about like a Sherringtonian neuron by neuron transmission of information, just like your airport example. But in the cortex and the higher level areas, and especially in a small patch of the cortex, I tend to feel like that the computation is not happening neuron by neuron, rather by just like working on all these network of recurrent units all together at the same time. So in that scenario, how do you think the small wordness is useful? Do you have any intuition on it? That would be really helpful for you.
Yeah, so this talk was very focused on single neurons, its single neuron tuning properties and connection statistics. Actually, most of the work in our lab focused on population dynamics because we record this large populations, and the other work that we hope to submit soon. It focuses on the dimensionality of the activity in the motor cortex and its relation to anatomy tuning and these distant dependencies. And what we see is a very high dimensional activity space in the motor cortex that sort of scales unboundedly with the number of neurons. So on top of this local network structure, we essentially see a lot of activity dimensions on the level of the population. And in addition to recurrency, a very easy hypothesis to make is that basically we are at layer 2-3. This is the autoassociative cortex. It receives a lot of inputs from other cortical regions. So these local neurons meet a lot of inputs from areas further away. So with whether this small world functionality of the hub neurons on the local networks, giving this high dimensional activity space that these neurons are embedded in, whether these neurons might essentially sort of participate in a more dominant activity dimension, and that suggests goes together with this observation that they are more correlated to the network, whereas a less connected neurons, sort of more private neurons, might maintain a less dominant dimension that still might do some specific computation, either in motor control or in specific reward processing. This is one speculation that we have, and I agree with you that this is not sort of neuron by neuron interaction that probably are the sort of the dominant motifs here. Yeah, sorry for the vague hand wavy answer.
No, but that's very insightful. So hub neurons essentially kind of linked to higher PCs, essentially.
Well, we didn't test it, but that's maybe one speculation that one can make.
I mean, it also reminds me of this idea that the brain is kind of preserving itself at a kind of a close to a critical point in a complex dynamic point of view. And you usually see this kind of scale-free networks that they follow this log distribution, which is like what you show here, that they have this hub nodes that are connected to a lot of nodes, like a preferential attachment network. And I think that's like a kind of it was a nice validation of that idea for me.
Yeah, I'm wondering, since you brought this, do you think of movement generation in the motor cortex as this sort of approach to the critical regime? Is I don't know what's your view?
Well, I mean, I see it as a general sense that it gives you the flexibility. I mean, you're in the somewhat middle ground, that you're flexible enough, but you're not chaotic, and you're also not just uninterestingly stable. And they usually see at this critical point that kind of dynamics emerge in these networks usually.
Yeah.
Yeah.
Yeah, great. So if there's,
Can I ask another?
sure, if you have time, Arseny.
Yeah, it's evening here. I'm completely free, so
here's another question from it.
Maybe a
Yeah.
final question. Thank you, Arseny. My question was, I think you mentioned that when you stimulate the neurons, it's in sort of different time points randomly, I think. And I was wondering, so if you're in a certain task, you're in a certain neural dynamic, and if you like suddenly stimulate in time point A versus time point C, I guess the population activity would sort of go into the perturbation in these two different time points would make the population go in different sort of neural states. And what was that decision based on? And do you think that it matters at what time point you do the stimulation? And would that change, like if you do that at rest, would that change this connectivity mapping that we get from this method?
Yeah, so that's a great question, and our sort of strong conviction is that we think that since we measure essentially activity, evoked activity in neurons to stimulation, which is supposed to be network state dependent, we think that essentially we are measuring some kind of a dynamical, a causal connectum, if you want. And whether some of these connections would be just observed always or would be state dependent, that's a very interesting question. That's something that we are that we're starting to look at it now. In this particular paper, we mapped the connectivity not during the behavior. So it was basically a spontaneous rest, if you want, epoch, where we measured the connectivity and then started the behavior. So we recorded the same neurons, but the connectivity was measured outside of the behavioral regime, partially in order to, because we needed enough time to make this connection, partially in order to sort of not contaminate the measurements by the ongoing behavior. What we see is that there is some relationship between the connectivity across different states. So we think at least some of these features would be sort of shared, but even within the behavioral state, and that's sort of directly relates to your question. The population activity before, during, and after movement is different. And when we measure the structure, and this is without connectivity, when we measure the structure of the covariance between the cells at these different epochs, this structure completely changes. And also the structure of the interactions across areas. As I mentioned in this other recordings we record about 10 different areas simultaneously, so we can measure their interactions as well, not only on the local, but also on the meso scale level. So there is a lot of change in the covariance during these transitions to movement and following the reward consumption. So I would sort of bet that at least some of this connectivity motifs would be different, and that would be also related to sort of different information flow patterns locally.
Thanks. That would be a nice follow-up paper. Thank you. Thank you. That was amazing. And thanks for answering all the questions. Yeah.
Thank you.
Thank you very much for the invitation, and I will be happy to interact in any other forum if there is an interest to do maybe a joint lab meeting someday. I would be very interested. We are because we work on rodents and we sort of follow the human and primate literature quite a bit, and sort of interested in this broader perspective on the motor cortex function. So thank you for the opportunity, and I'll be happy to interact more.
Yeah, likewise. Yeah. Have a great evening.
Thank you, Arseny. Have a great evening.
Have a good day. Bye-bye.
Bye.